Welcome once again to Population Genetics and Evolution, Unit 2, Session 1, Polygenic Inheritance. And by the end of this session, you should be able to explain polygenes, state and explain different types of gene action, distinguish between continuous and discontinuous variation, and draw graphs to illustrate them. A segment of DNA occupying a specific locus or position on a chromosome which constitutes the basic unit of heredity is called gene. And genes act by directing the synthesis of proteins, which are the main components of cells and are the catalyst of all cellular processes. Physical traits such as the shape of a plant's leaf, the coloration of an animal's coat, tongue rolling, height, weight, intelligence, and the texture of a person's hair are all determined by genes. So what are polygenes? Polygenes are so-called multiple factors, multiple genes, minor genes, or quantitative genes. They are genes whose individual effect on a phenotype is too small to be observed, but which can act together with others to produce observable variation. Polygenes constitute a group of non-allelic genes that together influence a phenotypic trait, with each gene expressing small additive or cumulative effect. The precise loci or identities of the non-allelic genes are usually unknown. However, advances in statistics and high throughput DNA sequencing techniques made it possible for researchers to locate candidate genes for the trait. The position on a chromosome where a gene is located is called a locus. Polygenic locus is any individual locus associated with a system of genes responsible for the genetic component of variation in a quantitative character. Polygenic locus may be either single or complex genetic locus. Polygenes are also known as quantitative trait loci, which control quantitative traits as opposed to qualitative characters with monogenic or oligogenic determinism. Polygenes are associated with genetic loci controlling characters such as color of the skin, height, weight, etc., which are expressed as continuous variation leading to normal distribution of the phenotypic value in a population. Since gene effects do not always fall into clear-cut categories and quantitative traits are governed by genes with small individual effects, they are often described by their gene action rather than by the number of genes by which they are encoded. Gene action is a functioning or expression of genes, hereditary units, in determining the structural and functional characteristics of an individual, that is, its phenotype. There are 4 types of gene action: additive dominance, epistatic, and overdominance. It should be pointed out that gene action is conceptually the same for major genes as well as minor genes. The essential difference being that the action of a minor gene is small, cumulative, and significantly influenced by the environment. However, major genes control clear-cut traits that are classified as discontinuous variation and are not often significantly influenced by the environment. The effect of a gene is said to be additive when each additional gene enhances the expression of the trait by equal increments. Example, if one dominant gene adds 1 unit to a trait, the effect of recessive AA, recessive BB equals 0. Dominant A, recessive BB equals 1. Dominant A, dominant B, recessive B equals 3. And dominant A, dominant B equals 4. For a single locus, A dominant, A recessive, the heterozygote will be exactly intermediate between the parents, that is, AA dominant equals to A dominant, A recessive, that's co-dominant, that's 1, and AA, that's recessive, giving us 0. That is, the performance of an allele is the same irrespective of other alleles at the same locus. This means that the phenotype reflects the genotype in additive action, assuming the absence of environmental effect. Dominance action describes the relationship of alleles at the same locus. Dominance variance has 2 components: variance due to homozygous genotype and variance due to heterozygous genotype. Dominance effects are deviations from additivity that make the heterozygote resemble one parent more than the other. When dominance is complete, the heterozygote is equal to the homozygote in effect. That is, dominant A, recessive A equals dominant A, dominant A. The breeding implication is that the breeder cannot distinguish between the heterozygous and homozygous phenotypes. Overdominance gene action exists when each allele at a locus produces a separate effect on the phenotype and their combined effect exceeds the independent effect of the alleles. That is, recessive A equals 1, dominant A equals 1, and co-dominant A, that's recessive and dominant equals 2. From breeding standpoint, the breeder can fix overdominance effect only in the first generation, that is, the first filial hybrid cultivars, through apomixis or through chromosome doubling of the product of a wide cross. Epistatic effects in qualitative traits are often described as the masking of the expression of a gene by one at another locus. In quantitative inheritance, epistasis is described as non-allelic gene interaction. When two genes interact, an effect can be produced where there was none. Example, dominant A, recessive B equals 0. Recessive A, dominant B equals 0, but dominant A, dominant B equals 4. Differences in genes and their expressions coupled with environment bring about variations among organisms of the same or different species. Thus, genetic variation is due to mutation and recombinations, as with regards to figure 1. So, the kind of gene interactions between alleles of a single gene controlling a character are referred to as allelic or intra-allelic interaction. Non-allelic or intra-allelic interactions also occur where the development of single character is due to two or more genes affecting the expression of each other in various ways. Indeed, Mendel and other workers assumed that characters are controlled by single genes, but it was later discovered that many characters are controlled by two or more genes, which affect the expression in various ways, leading to the modification of the typical dihybrid ratio of 9:3:3:1 or trihybrid ratio of 27:9:9:9:3:3:3:1. Genetic variation refers to small differences that exist between individuals of the same population, which can be described as being either discontinuous or continuous. It's where individual phenotypes occur as discrete and fall into a number of distinct classes or categories based on characters that cannot be measured across a complete range. The characteristics are discrete without intermediate forms. You either have the characteristic or you do not. Blood groups are a good example. You are either one blood group or another. You cannot be in between them. That's with regards to figure 1 in your textbook. Such data are termed as discrete or categorical data. Chi-squared statistic is used to analyze such data. Discontinuous variation is controlled by alleles of a single gene or a small number of genes. The environment has little or no effect on this type of variation. Characteristics of discontinuous variation. A, they have distinct categories into which individuals can be placed. B, they are qualitative in nature with no overlap between categories. C, they are controlled by one gene or a few number of genes. D, they are largely unaffected by the environment. So this is the type in which there is a continuum of characters or traits with intermediate forms expressed as a complete range of measurement from one extreme to the other. Height is an example of continuous variation. Individuals can have a complete range of heights, for example, 1.6, 1.61, 1.62, 1.63, etc. meters high. Other examples of continuous variation include weight, handspan, foot size, milk yield in cows, etc. Continuous variations controlled by polygenes or many genes known as polygenic inheritance and is often significantly affected by environmental influences. Milk yield in cows, for example, is determined not only by their genetic makeup, but is also significantly affected by environmental factors such as pasture quality and diet, weather, and the comfort of their surroundings. Do you know your body weight? When body weights of a large group of people are plotted as a histogram, the result shows a typical bell-shaped normal distribution curve with a mean, that's average, mode, largest value, and median, that's central value, all being the same in figure 2 in your textbook. So the characteristics of continuous variation, they have no distinct categories into which individuals can be placed. They are quantitative in nature with overlaps between categories, controlled by a large number of genes or polygenic. They are significantly affected by the environment. In review, genes are responsible for hereditary characteristics of organisms. Polygenes control quantitative characteristics. They have small but additive effects that are expressed phenotypically in a continuous variation. The expression of polygenes is significantly influenced by the environment, which brings about continuous variations in a population. On the contrary, single or major genes control characteristics that are expressed as discontinuous variation. Continuous variations are qualitative and discontinuous variations are quantitative in nature. However, variations can due to genetic and environmental.